Welcome back to my channel, and I am Anukampa going to discuss the third episode of genetics. In today's episode, I will not discuss about the dihybrid cross but a step before the dihybrid cross, and that concept is about the gamete formation. Generally what happens whenever we head towards the concept of dihybrid cross, then many of us feel so many problems while forming the gametes. So today's episode is totally focused on the gamete formation. So watch this video till the end because there are going to be so many concepts in a very easy manner. So before heading towards the gamete formation process, we need to know that what is gamete actually mean. So gamete is the reproductive cells. About the reproductive cell, we will get into the details when we will talk about the reproduction topic. So right now, you just need to know that reproductive cells are the cells which are actually involved in the reproduction, means in the zygote formation. So there is going to be a reproductive cell of male, which is generally termed as in human sperm, and there is going to be a reproductive cell of the female that is termed as ova in human, and it is going to fuse together to form a zygote. And as each and every individual in humans, in us, is going to be diploid, so these gametes or reproductive cell needs to be haploid in nature, because when these haploid cells will fuse together, then they will be able to form the diploid cell. So this is the complete information that is required whenever you are going to understand the gamete formation process. Now, heading towards the process. To talk about the dihybrid cross, then as in one of my video I have already explained that dihybrid means when we are considering two traits and we are observing the hybrids formed by those two traits. When we talk about this gamete formation process, we consider the dihybrid cross, that is, we are considering the two traits. Now whenever we are considering two traits, so now here I am considering these traits as the one trait is height and another is hair color. Trait means characters, right? So this first trait, which is height, is shown by the letters of alphabets capital T and capital T. That capital V is trying to represent the trait that is hair color. So now if we will consider these two traits, so this is the case 1 where these two traits in the parental genotype, this is the parental genotype, in this condition, these traits are in homozygous dominant condition. Now, whenever you want to form the gamete in this condition, so it is very easy because this is in the diploid state. What do you mean by diploid state? When the alleles are in pair. Here the T is in pair, the B is in pair. So they are having the duplicate. Now we will just make half of it, that is the haploid. So diploid means if there are two, they are in pair. Now we will segregate them, so one T and one B from here. So we need to have both the traits in the gamete, but for both the traits it is going to have only and only single allele, that is one gamete. Now there is not any other form of gamete that is going to form over here. So this gamete formation is very easy with everyone. Considering the case number 2. In this case, this one is going to be in heterozygous condition, whereas this one is again in the homozygous condition. So now in this case, I will tell you another concept. That concept is whenever there is this kind of condition, means there is some heterozygous condition is present in the parental genotype, then you have to go through one formula. That formula is to know the number of gametes that can be formed. This formula is number of gametes should be equals to 2 raised to the power n, where n is equals to the number of heterozygotes present in parental genotype. This is the parental genotype, and as you can visualize over here that only one heterozygous condition is there. Only one trait is in heterozygous condition, another is in homozygous condition. So here the value of n for this case is going to be 1. So for this case, if you will keep the value in this formula, then what you will receive, that 2 raised to the power n, that is 2 raised to the power 1, is going to be equal to 2. So in this case, we are going to have 2 gametes. The number of gametes are going to be 2. This will help you in confirming that now you have to only form 2 types of gametes in this condition. Now let's understand how to form those 2. We will do. Now again, this is diploid condition. I have to convert it into the haploid form. So first I will consider capital T and I will take any of the capital B from here. So this is one form of gamete. Another, I can take small t from here and I can take any of the capital B from here. And as we only need 2 types of gametes, so these are the 2 gametes. So this one is also easy, but it is little bit complex than the earlier case that we have discussed. Coming on to the case number 3. Now let's understand case number 3. For case number 3, now we are having this high trait in homozygous condition, but just opposite of the case number 2. Now the hair color is in heterozygous condition. So again, the number of heterozygotes in my parental generation is only 1. Again, keeping n here as the value, we will get number of gametes as 2. And when we will form the gamete, then what we will observe, we will take capital T from here and one of the like capital B from there. This is one gamete. Again, we can consider any of the capital T and small b from here because capital B we have already taken. So these are the 2 forms of gametes for this case number 3. The last case, that is case number 4, and which is the most important case where the students face problem. That is when both the traits are in heterozygous condition. In such cases, now you can check that there are 2 heterozygotes. This one is also in heterozygous condition and this one is also in the heterozygous condition, right? So now the value of n is going to be 2. So now when you will keep the value of n equals to 2, then 2 raised to the power 2 means 2 into 2 means it is going to be 4. So now here we will find 4 types of gametes. Okay, little bit tough, but it's not tough. Just keep watching this video. For this condition, you can apply 2 methods. The first method is, as we were doing previously, we will take capital T from here, capital B from here. We will take capital T from here but small b from here because there are 2 combinations that can be observed. So I have combined this with this and this with this. Now I will take small t from here, I will combine it with capital B. I will again take small t and I can combine it with small b. So there, these 4, we have to form 4 gametes and these are those 4 gametes. But sometimes students get confused. Why this capital T is occurring twice? It is occurring twice because it can combine with both of them. Probabilities are for both the combinations. But if you are getting confused, let's talk about the another way, and that is the fork method. In this method, what you have to do, take any one of the trait and write it like this, capital T and small t separately. Then form a fork in front of it. I have formed a fork. Now in fork, write the alleles of the second trait, that is capital B and small b, again capital B and small b. Fine. Now you have to read it in the from upward to downward manner, like capital T, capital B. So one gamete is capital T, capital B. Again capital T, small b. So next is capital T, small b. Now read it in this direction, small t, capital B. Then again read it in this direction, small t, small b. And these were the 4 gametes that we have observed by the first method also. And we need to know only 4 gametes in this condition. So this is the simpler way. So these are the 2 ways by which you can go for the gamete formation in a dihybrid cross. Students also get confused when they go for trihybrid cross, and there you have to go for the gamete formation. But don't worry. Just continue watching the episodes that I am going to upload day by day, and you will observe that these concepts are going to be very easy for you to understand. Okay, we have discussed about 4 cases of gamete formation. We talked about what is gamete. Then we discussed that the gamete is actually formed by the parental genotype conditions and forms. So there were 4 forms in a dihybrid cross, and we discussed all those 4 forms and the methods that we can use for the gamete formation. In case you are feeling any question or any doubt in this discussion, you can write your question in the comment section. If you have liked this video, then do a thumbs up and like, share, comment and subscribe my channel.